Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International GCSE Biology Paper 1B for January 2022. This is the Part 3 video. I did 2 more videos and I'll put their links below the description box of this video. Let us begin with question 8. The rate of photosynthesis is affected by different factors. One factor is the concentration of carbon dioxide in the air. The percentage of oxygen in the air is 21%, and this is equivalent to a concentration of 210 parts per million. The percentage of carbon dioxide in the air is 0.04%. They want you to calculate the concentration, or this concentration in parts per million. We can begin by looking at what occurred. If they said 21 over 100, which is 21%, is equivalent to 210 parts per million, it means they multiply this by 10 power 6 to convert it to the other concentration. Therefore, we can do the same thing. For CO2, we can say 0.04 divided by 100 times 10 power 6 should give us 400 ppm, which is the desired concentration here. The next part says, the graph shows the effect of increasing the concentration of carbon dioxide in the air on the relative rate of photosynthesis at different temperatures. So here on the vertical axis, we can see there is relative rate of photosynthesis, and here we can see the concentration of carbon dioxide. As the CO2 concentration increases, the rate of photosynthesis increases. Also, as temperature increases, the rate of photosynthesis increases. We can use this example here, the concentration of 0.08. Drawing a vertical line at all temperatures, at this concentration we can see the higher the temperature, the higher the rate of photosynthesis. 35 degrees has the highest, and then 5 degrees has the least rate of photosynthesis, even at the same concentration of CO2. Let us move on. Here they say, describe the effect of increasing the concentration of carbon dioxide on the relative rate of photosynthesis at 5 degrees Celsius. So I'm going to go back to the graph and look at 5 degrees and see what we can see. So moving back here, at 5 degrees, we can see this is what we're talking about. We can see that the higher the concentration of CO2, the higher the rate of photosynthesis until a specific concentration. So we can see until about here, then we can see the rate is going to level off. So we can see concentration increases, or actually you could look up here and see when it completely levels off, it's about here, about that concentration. Or let me just remove it so that you can see. About a concentration of that part, about 0.1, 0.9, and so on, we can see the rate becomes almost constant about that point there. That is when about closer to 0.1% the concentration of CO2, so we can see the rate becomes constant. Initially, it's going to be increasing, and then it becomes constant. So let's go back here. I said increasing the concentration of CO2 increases the relative rate of photosynthesis. It reaches a maximum at 0.1. I said it reaches a maximum at 0.1 because based on the information given, let me try to erase so that you can see clearly. It looks like this is the maximum obtained rate of photosynthesis, or at that point where the concentration is 0.1, we have the highest rate of photosynthesis or relative rate of photosynthesis. The next part says, describe how the effect of increasing the concentration of carbon dioxide on the relative rate of photosynthesis changes when temperature is increased. We know that increasing the temperature increases the kinetic energy the particles have, therefore rate of reactions will always increase. So here I said, increasing the carbon dioxide concentration increases the relative rate of photosynthesis at all temperatures. And then we say the steepest increase in the rate of photosynthesis is at 35 degrees. That is evident from the graph. The highest increase was at 35 degrees. And again, remember, this is asking us to describe how the effect of increasing concentration of CO2 and the relative rate of photosynthesis changes when temperature is increased. The higher the temperature, the higher the relative rate of photosynthesis. From the graph, that was evident. Then here, lastly, they say explain the effect of increasing the temperature from 5 to 35 on the relative rate of photosynthesis. This question is asking us to explain, so we have to show, we talk about what change has occurred and the reason as to why that change has occurred. Now, this is where we talk about the kinetic energy. We know the higher the temperature, the higher the kinetic energy the particles or substances that are reacting are going to have. Most of these reactions are enzyme controlled, so we know that when the temperature is increased, the enzyme or the rate of reaction is going to increase because the enzyme and the substrate will have more frequent collisions. So here I said increasing the temperature increases the relative rate of photosynthesis. This is what is happening. Then we can give a reason for that. A higher temperature provides the right kinetic energy for molecules. These molecules will ensure that the molecules collide frequently, or they have frequent collisions between the enzymes as well as the substrate. And then the temperature is a limiting factor at 5 degrees. Remember, we say the higher the temperature, the higher the rate. At 5 degrees, temperature is limiting. And then at 35 degrees, of course, we're going to have a higher rate due to more frequent collisions between the enzyme and the substrate. So here I said increasing the CO2 or CO2 concentration has less effect on the rate of photosynthesis. This is if temperature is high enough, then the rate is going to be high enough. However, at 5 degrees, because temperature is limiting, no matter how much the CO2 concentration is increased, the rate of reaction will not increase significantly. So this is linked. The temperature is the limiting factor at 5 degrees because increasing CO2 has less effect on the rate of photosynthesis. This is at that temperature. So when you increase the temperature and increase the CO2, then you have a higher rate. So let's continue to the next part. Scientists who carried out this study concluded that the effect of increasing the concentration of carbon dioxide on the rate of photosynthesis of a plant is dependent on temperature and also on the minerals that the plant can absorb. They explain how lacking a named mineral might affect plant growth. The named mineral I chose here was a nitrate. The nitrates would lead, so lacking nitrates would lead to less amino acids. And since amino acids are used in making proteins, which are used for the growth or making structural component of the plant, if there are fewer nitrates, less amino acids are going to be present, less proteins, and therefore there is going to be less growth, or you say stunted growth. You could also choose magnesium. Since magnesium is used to make chlorophyll, lack of magnesium will mean lack of chlorophyll, so we will see yellow leaves. The next part says, explain how a named factor can affect the rate of photosynthesis other than carbon dioxide concentration, temperature, and minerals absorbed. I chose light intensity because, as you can see, availability of light intensity provides the energy required to cause opening of the stomata. So if there is right amount of light intensity, then there is going to be opening of the stomata, and then gases can be trapped in, or they can come in order for photosynthesis to occur properly. You could also choose water because water is a substrate in photosynthesis. That could be a limiting factor as well. So let us continue to question 9. Question 9. Alkaptonuria is an inherited condition caused by the presence of recessive alleles. State what is meant by a recessive allele. A recessive allele is the allele that is only expressed if it's present in homozygous recessive form in the genotype. So B says Alkaptonuria is first diagnosed in children when it is noticed that they produce very dark urine that turns black when exposed to air. A woman and a man do not have Alkaptonuria. They have a child who has the condition. The woman and the man are expecting a second child. Draw a genetic diagram to show the genotypes of a woman and the man, the gametes they produce, and the possible phenotypes, the genotypes of the second child. So phenotypes and genotypes of the second child. So for me, I said using A and A as possible alleles. So the parent's genotype is going to be heterozygous because you realize the parents did not have the condition, but they had a child. They did not have a condition, but I think they had a child who had the condition. So it means they were both heterozygous. So one heterozygous father, the other heterozygous. So the total gametes that are going to be there is a capital A and a small a. So this parent and that parent produce that, and that. This is produced from that and that and that produce that. So that and that produce that, and that produce that. So these gametes are going to produce these genotypes as offsprings. So the offspring genotype is going to be a capital AA. This is homozygous dominant. This is heterozygous, and then homozygous recessive. So the offspring phenotype is a 3-to-1 because we have 1, 2, 3 who are going to have no symptoms, while we have one who is going to have Alkaptonuria. So that is what they asked us for. Continuing on, they say calculate the probability that the second child is male and does not have the condition. Remember, if they say and in probability, it means you have to multiply. So here what we're going to do is we're going to find the probability that the child is male. which is always a half, times the probability of not having this condition. The probability of not having this condition, let's go back to the answer we got previously. Remember, for these parents, 3 children will not have the condition, so 3 out of 4. It means the probability is 3/4. So let's go back here. You can see it's going to be 3/4, while that is a half. So a half times 3/4 gives you 0.375, which is the answer here. The next part says, Alkaptonuria is caused by the body being unable to break down the amino acids tyrosine and phenylalanine. This leads to a buildup of a toxin that causes damage to joints and tendons and can lead to heart valve damage in later life. A new drug treatment is being tested that can slow the damage to the joints and tendons. Scientists selected 40 adults who all had Alkaptonuria. They placed each patient into one of the 2 groups. One group was given a drug treatment and the other group acted as a control. The scientists then compared the symptoms of the patients in each group after 3 years. Describe what is meant by a control group. So a control group is the group that did not receive the drug. So they did not receive the drug. They were just there for comparison of the results when 3 years later passed. So basically, they did not receive the drug. The next part says, the table compares the control group with the drug treatment group. It shows the number starting and completing the trial and those showing harmful effects. It also compares improvements into symptoms of Alkaptonuria. So here we can see the results in this table. The number of patients starting the trial were 20 for each. Number of patients completing the trial, here we had 17 and 16. So it means if some people left the trial, then they're decreasing the number and they're making our results not as consistent. The number of patients showing adverse effects for control group was 0. Here there were 2. And then the number of patients who died, 0. Here we had 1. So we want to know if the drug killed the person or they had other issues. Decrease in time taken to stand up and walk 3 meters in seconds. So it's going to be, we can see there is a decrease. An increase in distance in meters walked in 6 minutes. There is that and that. So they say evaluate whether the new drug should be recommended as an effective treatment of Alkaptonuria. Here there is a yes or a no. I said yes because there is a control group. So here having a control group is very important. So the study can be valid because there is a control group, meaning the changes that are going to be observed here are going to be due to the independent variable that was varied in this experiment. So the results showed improvement in activity. For example, there was a quicker time to stand up, and then there is improvement in distance walked. So we can see there is some positives. However, we may not recommend this because the sample size was really small. They actually used only 40 people, and if you ignore the control group, they just used basically 20, who turned out to be 17. So that is a very, very low number for these results to be reliable. So a small group was used, more people should be tested. So meaning the experiment should be repeated in various studies. And not all people finished the study, so that is a problem as well. Some people experience side effects. That is a problem. There was one person who died in the drug group, so that is also problematic. It means maybe the drug can cause some problems that they need to investigate. There is no information given about the ages, the masses, the sex, the activity, the health conditions of the people who were used in this experiment. And as you know, men and female could react to a similar drug in different ways due to their different body mechanisms. So also people who have different ages or people who have different pre-existing conditions, they could react to different medications differently, and that could be problematic. So next we see, other scientists have suggested that eating fewer proteins that contain tyrosine and phenylalanine would reduce the symptoms of Alkaptonuria, suggest why eating fewer of these proteins may be difficult. Now, this is going to be difficult because how can you distinguish which foods have these and which foods do not? Most of these foods in nature could have them, so it's going to be really crazy and difficult. So I say patients may not know what foods contain these, the tyrosine and phenylalanine, so it's going to be very hard for them. And also they may not know if these substances are contained in most foods or most proteins. So it's going to be a painstaking process for them to find out which is which. And some of them will end up not eating proteins at all, which is going to affect their growth and repair. So that is going to be a problem as well. This brings us to the end of question 9. Let's continue to question 10. Question 10, Farmers sometimes use biological control to reduce the damage of their crops caused by pests such as insects. Which of these is an advantage of using biological control over chemical control? Biological control is specific. It only targets specific pests, so it's going to be advantageous in that way. The next part says, aphids are tiny insects that have very sharp mouth parts. They push these mouth parts into the phloem found in stems, and they then feed on the phloem contents, as you can see here. So they say, name 2 substances the aphids obtain from the phloem. The phloem contains sucrose, and it contains amino acids. So sucrose could be used for energy, and the amino acids could be used in the production of protein. So these are contained within the phloem, and they will obtain them from in there. Here they say, explain how aphids feeding from the phloem of crop plants can lead to reduction in yield. This reduction in yield, we're talking about the yield of the plant. So here, since we said the aphids are going to get the sucrose and amino acids from the phloem, so there will be less sucrose and amino acids available to be stored within the plant or for the plant itself. So the plant will have less respiration taking place, and with less respiration, there will be less ATP being generated. And since ATP is required for some processes that occur within the plant, there will be less active transport. So active transport requires energy. There will also be less growth of leaves, bulbs, and stems. These require proteins. So amino acids are removed or they're decreased, so there will be less proteins made. So there will also be less photosynthesis, since photosynthesis requires energy, and this energy is going to come from the ATP production, basically. There will be less nectar, since nectar is a sugar that could be made, so there will be less nectar. And since nectar is required for insect pollination, so there will also be less insect pollination. So you can write as much as possible or as much as you want. They will all award you the marks. This was just 4 marks, so any 4 points you get the 4 marks. I'll continue to the next part. Silverflies and hoverflies are 2 species of insects whose larvae feed on aphids. Scientists investigated the feeding behavior of these species in a laboratory experiment. This is the scientist method. They place a single silverfly in a container. Now, this is very important. Remember, single. A single silverfly in a container, and they place a single hoverfly in a separate container, and they keep the containers at 12 degrees. They put 30 aphids in each container, and then they count the number of aphids consumed each day for several days. They determine the mean number of aphids consumed per day, and the scientists repeat the experiment, or they repeat the method at 2 higher temperatures, and then the graph. Here is a graph that shows the scientist results, basically. So I tried to write in some values that will match with the mean numbers of aphids consumed per day to make it easier. So we have the hoverflies, which are those, and then the others are silverflies. So I tried to calculate, I think you can see calculations of important data. This is from the next page, so I'll come back to this part. However, let's just go, I'll come back to this part. So here they say the scientists concluded that the hoverfly is the most effective biological control agent for aphids. So they ask you to discuss the scientist's conclusion, referring to the information in the graph and in the scientist's method in your answer. So here we're going to refer to the information. So, and again here I had a yes or no, so agreeing with the scientist or disagreeing. So I said yes, it is conclusive because the hoverflies consume more aphids at all temperatures. If you go back again here, you can see mean number of aphids consumed. The hoverflies are consuming more at all temperatures. So it's showing us that maybe they are better at this biological control. Therefore, hoverflies should be used. And also we saw the greatest difference is 3 times at 12 degrees Celsius, and at least the least one is 1.9 times at 18 degrees. And again, to take you back, you can see at 12 degrees I saw there is 3. At 15 degrees we have a 2, and here we have a 1.9. So this is 1.9 times, this is 0.2 times, and this is 3 times. So I calculated it at that. So when we refer back, you can see these points I put 3 times at 12 degrees and 1.9 times at 18. And then yes, because both flies consume more at higher temperatures. So the higher the temperature, at least for the experimented temperature, the higher the consumption. However, the results are not conclusive because there is only one larvae that was used. more larvae should be used so that we can know. In real life, there is not going to be just one larvae, there could be many, and we need the data to depict what would happen in real life. So the data is less valid since the experiment was carried out in the lab. Results may be different in the natural habitat. That's correct, because lab conditions are more controlled than what will happen in real life. So the result, compare the results with other species of flies, not only hoverflies or silverflies. So maybe there could be others that are better biological control agents. So we have to work with more different types of flies and see the comparison. This brings us to the end of this paper. This was the third video, so please, thank you for being with us. Do not forget to subscribe. I'll see you in the next video. Bye-bye.